Good. Okay. Okay. Good. All right. You know, Ray, are you there? Can you hear me?
Yes, I can hear you.
Okay. We're going to start in about 1 minute, I think. I just wanted to make sure you were still there.
Thank you.
Okay. Um, all right. Give it 1 minute. Just waiting for the last people to filter in. Buffalo time is usually, 15 minutes past regular time.
Okay.
All right. Well, unless I'm just going to start in, I think, and I'm going to figure that people might come in as we start, but since this is just the introduction, it's kind of the part that people can miss and still enjoy the night. Thank you for coming. Welcome to Buffalo in the Public Sphere. This is Buffalo in the Public Sphere version 4. And these yearly events have showcased artists and their in-progress work from UB's Coalesce Center for Biological Art for the last 4 years. For those of you who don't know, Coalesce is dedicated to advancing new humanistic tools and ethical methodologies to correspond with a rapidly growing biotechnological sector. Coalesce is a hybrid studio laboratory that facilitates creative engagement with the tools of the life sciences, primarily through artist residencies in our lab, UB courses, workshops, and exhibitions, such as an upcoming event with Torrance Space Theater here in Buffalo. And I have to say, I have to thank Hall Walls and John and Bill and Ed Cardoni. We've been collaborating now, I think, pretty much ever since Coalesce began, which is about 6 years ago. We've been hosting these events here, and they also are partners on our last NEA grant. And this series is even part of what we had planned to do with NEA all along. For that matter, then I need to thank the NEA down here at the all the way down at the bottom. So we received an NEA Artworks grant in 2019, and that grant has allowed us to really propel this Artist and Residency program ever since. Before then, yeah, anyways, so I have to thank the NEA for that. Before then, we were originally funded through a provost initiative for Communities of Excellence, and we are part of the Genome, Environment, and Microbiome Community of Excellence since its inception. I should also mention we are part of the LASER network. LASER network up here stands for Leonardo Art and Science Evening Rendezvous. Easier to say LASER. And Buffalo is one of more than 30 cities. I think now it's actually almost 50 cities across the world. And we're an official LASER host, which is an international series organized by the Leonardo International Sciences for the Arts and Art Sciences and Technology. From that, then, I thought I'd briefly introduce all the panelists today. I'm going to do this in about 2 minutes. The first presenter is Jens Hauser. Jens Hauser is a Paris and Copenhagen-based media studies scholar, author, and art curator focusing on the intersections between art and technology. As a curator, he's organized over 30 international exhibitions and festivals, focusing on ideas like biomediality, skinterface, liveness, and finally microperformativity, a term that he has coined to describe and theorize unique attributes and contributions of these very visceral works like these. And then I'm going to swap the order a little bit, I think. The second presenter, if this is okay with you, Ray, is going to be Ray Yuping Su. Ray is an artist and educator from Taipei, Taiwan, currently based in New York and teaching at the New School. Her project and question that she comes to coalesce with is how many citizens are living in this body, which addresses the notion of the body itself as a place of immigration and colonization as well as acculturation, and uses traditional foods alongside microbiology techniques of knowing. The second talk is by Roland van Dierendonck and Julian Standon, and they're collaborating on a project called Feel Your Fermented Microbiome, or Feel Your Fermented Biome, sorry. And it uses haptic interfaces to translate their changing gut microbiome. Roland is an artist and PhD candidate at the Lab for Living at the Sheffield Hallam University, combining haptics touch with microscopic life. And Julian is an Australian artist, designer, curator, researcher, and educator who lectures at the Interface Cultures and Media Design teaching programs at the University of Arts and Design in Linz, Austria. And then finally, Tosca Terán is a Toronto-based multidisciplinary artist. Tosca founded Nanopod, a hybrid studio makerspace in 2005. Continually exploring new materials and tools, Tosca started collaborating artistically with algae, Fusarium polycephalum, and Mycelium. I might have mispronounced that, and I'm the art and bio guy. And Mycelium in 2016, translating biodata and non-human organisms into music. Her residency project, Symbiosis Dyssymbiosis, is an immersive mixed reality participatory experience working with living mycelium biodata sonification within a VR environment. I'm this close to the end. Each of these panelists is going to speak for 20 minutes, after which we'll be able to take questions or join in discussion with you and the audience. And again, really thanks for coming out and showing your support tonight. So thanks a lot.
Yeah, thank you, Paul. Thank you for this kind introduction and invitation again here back to Buffalo. It's always a pleasure to come back to a place which has a long history in relation to art and biomedia and activism criticality. It's one of the strong places, I think, in the world where such art has been growing over a kind of very long period of time. And I have the pleasure to open this squareless event on BioArt in the public sphere. And I'm very curious to get a deeper insight of the projects developed by the artists, researchers throughout this residency. So as an introduction, I will share some thoughts on alternative non-human animated agencies in art, which I've been increasingly addressing as microperformativity over the last years. And I want to introduce and put up for debate this notion of microperformativity, which I have been fruitfully working with, both as a media and art theoretician and as a curator, as a conceptualizing tool over the last years since 2003, in order to analyze and discuss these alternative animated agencies in biomedia art and performance art. And my hope is that proposing such a vocabulary in which performativity and agency is ascribed to systems of aliveness beyond anthropocentrically established criteria such as intelligence, consciousness, and language, may help also foster interdisciplinary encounters between art, philosophy, and the most diverse scope of technoscientific science disciplines when redefining allegedly self-understood but increasingly destabilized notions such as body, matter, and aliveness. So microperformativity is intended to describe the current and incremental trend both in the performative art practices and in performance art and in theories of performativity to destabilize human scales. By that I mean, of course, both spatial and temporal scales as the dominant plane of reference, and instead to emphasize biological and technological microagencies that, beyond this mesoscopic bubble of the human body, relate to the invisibility of the microscopic, to the incomprehensibility of the macroscopic. And to start, I want to describe the quite absurd circumstances, and maybe I can pass this around. It's already sold out, I'm sorry, but this is a journal we worked on for more than a year, precisely during the pandemic. Maybe you can pass this around, Paul. And so these were quite of absurd circumstances during which we worked towards a special issue for the journal Performance Research on microperformativity, co-edited with Lucy Strecker and published during the viral pandemic, but without having been initially motivated by it at all. And at this time this issue has been published, it seems surprising that viral agency had not appeared to be the central focal point of interest in the responses to our call, whereas a large range of papers dealt with bacteria and microbiota relating to the growing field of contemporary microbiome research and its appeal to cultural practitioners. And before COVID-19 affected and infected contributing authors, editors, and artists alike, the viral as material and metaphor was barely present in the reflection submitted, and we will come back to this a little bit later to this specific point. So we choose Blue Remix by Geneva-based artist Jan Marusic as a cover image for our issues for two specific reasons. First, because it subverts the tradition within which human phenomenological considerations are still rooted by displacing the focus from mesoscopic actions to microscopic functions, from physical gestures to physiological processes, and from stage diegetic theater time to real performative time. And in this mesmerizing 1 hour long performance that some of you might have seen, the former dancer Marusic stages a controlled biochemical choreography of methylene blue, which progressively seeps out all the orifices of his body, from the eyes, the mouth, the nose, eventually the pores of the skin, while internally body sounds are amplified and remixed, shifting the observer's attention from the physical to the physiological state. And second, this piece also relates to the frequent mode of microperformativity explored by artists, that is vegetality. And in the very polysemic sense of that term, not only meaning the quality or state of being vegetal, but also concerning this physiological phenomena that are commonly ascribed to plants, even in animal organisms. So the vegetative also alludes to this autonomic visual nervous system which regulates vital functions such as metabolic activity, breathing, and heartbeat, beyond categories that we take for granted like intentionality and subjectivity that are traditionally held central in human agency. And vegetal agencies, as an example, is also put forward in this physiological human-plant performance by Spela Petric, a 20 hours long motionless performance called scotopoiesis, meaning shaped by darkness. It involves vegetal artists both as co-performers and perceivers of the artist's own immobile art. And here the artist and germinating cress stand vis-à-vis each other, illuminated by strong light, and the continuous immobile shadow of the artist's body signals the plants to respond by changing their shape and color so that their bodies in turn produce a living imprint of her silhouette. So by abstracting light, the artist's shadows triggers a phenomenon called etiolation that results both in the paling and the lengthening of the cress's stem, which is then contrasted by the artist's own shrinking height due to the persistent stillness. And the deconstruction of apparent stillness was also at the core of a festival I curated in Munich at the Unsplit Festival in Germany. And for example, here are dendrites that are growing in real time out of vaporized mantle on a performer's body in crystal, a project by Russian artist Julia Borovaya, while in mass bloom explorations by Danish artist Tina Torpgaard, a human performer interacts with thousands of mealworms, non-human performance, so to slow down audiences to get in touch with decay, decomposition, and death. Under this dome, multiple cycles of life create constant changes to the landscape, while the mealworms digest polystyrene, human waste, cleaning up human waste, and create a unique soundscape. At the festival Applied Microperformativity in Vienna at the Angewandte Innovation Lab, we inquired how performative speech acts today unfold as spit acts, and how artistic methods can engage critically with technologies that exploit life on a microscopic and molecular level, merging bio and digital media. And think biopolitics and also necropolitics in relation to the dystopia of economy and the utopia of ecology alike. Human-animal relationships then, via the complexity here of immune communication systems and their microscopic agencies, are staged in this very well known performances and self-experiment by French duo A Orienté Objet, May the Horse Live in Me, that I co-curated with Capellice Gallery in 2011. Here, artist Marion Lavaljeanté was injected with compatibilized horse blood to express and to experience immune otherness in an act of transspecies blood brother or sisterhood. And the artist turned herself into a proverbial guinea pig, injecting herself over the course of months with horse immunoglobulins to develop tolerance to that horse immunoglobulins that would bypass the defensive mechanisms of her own human immune system, enter her bloodstream to bound with proteins of her own body, and then, as a result, impact on her own body functions of the endocrine system. And after the transfusion, Marion Lavaljeanté on stilts performed the communication ritual with the horse before her blood was extracted and freeze-dried. And this risky undertaking alludes to the possibility of healing autoimmune diseases using foreign immunoglobulins as therapeutic boosters, but the performance also was conceived as a continuation of the centaur myth, that human-horse hybrid, which, as an animal in the human, symbolizes the antithesis of the figure of the rider, who, as a human, dominates the animal. And more recently, interestingly, A Orienté Objet has carried out microbiome-based art experiences, portrait in our issue on microperformativity, consisting of grafting the rich microbiota of a pygmy inhabitant of the African primordial forest onto the artist, to potentially learn to feel, as a real physiological experience, the forest environment thanks to the transplant of an internal ecosystem in her environment. So traditionally, performance has been about the encounter between an artistic entity or presentation and its perceivers. Such co-corporality, as I call it, presupposes at least two types of bodies which establish a uni or bio or multi-directional affective connection. And today this requires to rethink to think of performance and performativity in terms of other than human agency, both biological and technical, and second, to redefine what a body is. Because in Latin, a body is just originally a corpus, and this means organized physical substance. And the term body does not at all imply any scale or nature, and is not anthropomorphic by definition. So we currently witness an attempt to escape the scales represented in this famous drawing by Leonardo da Vinci, the Vitruvian Man, in which the workings of the human body in its ideal proportions are visualized in order to establish analogies for the logics of architecture or other disciplines for the universe at large. And what today, despite Leonardo's intention, may be seen as anthropocentrism has come under critical and also humorous scrutiny, subverting this Vitruvian Man in the light of other metrics and logics that are, for example, molecular or microbial. So subversions question the human scale as the crucial reference point, and they also continue the idea of the three alleged wounds to human narcissism, in German die drei Kränkungen, earlier described by Sigmund Freud. First, Copernicus' discovery of heliocentrism as the first blow to humanity's narcissism and a critique to the idea of centralism as such. Then, of course, Darwin, who relegated the anthropos to just a descent from the animal world, and then, of course, Freud's own psychological explanations that the human ego is not even master in his own house, while today the microbiome is said to be influencing cognition. So probably these wounds need to heal very quickly in these times of anthropogenic effects on the ecology and the increasing technological potential to impact on living bodies at all scales. So what does art do, and what has art done in relation to aliveness in the past? So the creation of lifelike appearances has been an ever-recurring historical feature in art, but has been traditionally occupied with images, and also the ontological paradox that representations made of dead matter can indeed seem animated. And since the earliest anthropomorphic statues, myths of vivification have surrounded artifacts by the artist's hand. And the animation of malleable matter stands in a long tradition, Pygmalion and so on. So By the means of form, material, and process, aliveness has been staged and artists imagined, represented, mimicked, then simulated, and quite recently manipulated living beings and systems for real, since genetics, tissue engineering, DNA chips, so-called biobricks, and so on have entered the repertoire of a resolutely experimental form of art. So generally we have three categories we are dealing with, in which aliveness is being addressed. So first, of course, the traditional way of representation and concept-based traditional contemporary art made of that matter. Secondly, presence, process, and phenomenon-based dry media art with robotics and informatics, hardware and software. And third, likewise, presence, process, and phenomenon-based wet media art that employs biotechnology. The latter of two are relevant here with regards to how the media arts selectively stage life. So what we see is actually a trend from life to life, life with a V to life to an F, and from performance to performativity. So in the light of cybernetics and robotics, historically, artists have been very inspired by the simulation of so-called emergent intelligent behaviors, such as Gray Walter's turtles, these robotic vehicles equipped with sensors, able to explore the environment. And in the light of that, a whole genre of media art installations emerged, famously Ken Rinaldo's interactive and reactive sculpture Autopoiesis, or even further, the self-repairing robotic chair by D'Andrea and Dean that expresses, obviously, artificial aliveness just through movement, behavior, and learning, but not resembling any kind of organism at all. Aliveness, however, is staged in a very different way in two of unbalanced synthetic biology-based piece, Pigeon d'Or, attempting at making pigeons defecate soap by material modifying the metabolism of bacteria occurring in the gut with the help of biobricks. Or think of Wim Delvoye's defecating digestion machines called cloacas, where functional organic metabolic processes between enzymes and gut bacteria are enacted just to produce excrement. It's a definitely performative installation, and visitors often applaud as its droppings fall. So in that light, we need to scrutinize which characteristics of aliveness selectively put their emphasis on, artists put their emphasis on, since their choices also serve as indicators of the philosophical and technoscientific context within which they operate. Is it enough, for example, to identify a single body as being alive? Because in biology, the concept of life implies a transgenerational preservation and evolution. It deals not only with the structure and behavior of individual organisms, but also with its integration in subordinate ecological systems. So a very complete definition of life as manifesting a sum of characteristics, including some of which also we can find in the inanimate world, is proposed here, just the next example, by Bernhard Rentsch in what he calls an epistemology-based biophilosophy. And I'm not going to read out this whole definition, it would take 2 minutes, but it's a question of organization, cellularization, the notion of activity, metabolism, that it has to regulate, that it has to be irritable, that means that it has to have some possibility to de-evolve, to mutate, and also to have at a second order level a kind of psychic emergence. So while these criteria are adding up, we observe an apparent increase in complexity from the top to the down, but we may wonder how many of these complex characteristics are also fulfilled by lower level organisms or agencies. So the question is, which characteristics of the living are being emphasized by artists, when, how, and why? So practitioners of, let's say, dry robotic media art may emphasize activity, regulation, irritability, and let's say artists, those with an interest more in digital simulation of populations, but in non-biological media, they may focus on reproduction, on evolution or mutation, and practitioners of wet biological art may insist that their displays deal with metabolism, with dynamic stability, and the very protein-based materiality of the displays. So these preferences in turn also reflect onto the chosen art media with which the agents of aliveness are being coupled. And the very good examples of how these criteria can be combined, and I think we can today witness some of correspondences with the artist projects that we are going to hear about, is the so-called Posthuman sound piece, Self, by Austrian artist Guy Benari, featuring, quote, a rock star in a petri dish, playing together with a human jazz musician. And here, lab-cultured spiking biological neurons are generating sounds connected to electronic machinery, a kind of neural synthesizer. And this piece conceptually is of a certain particular interest because it combines in a very explicit sense performance and performativity. And these are not synonymous. So while in the notion of performance, this puts an emphasis of presenting something to an audience, mostly via human presence, the notion of performativity highlights the execution of whichever action or process. And here, for instance, the spike in nerve cells in a scientific apparatus, while the main purpose of this non-human performativity is not the encounter with an audience. And for this reason, this piece is also analyzed in our issue on microperformativity by Chris Salter, who is also the author of Entangled, Technology and the Transformation of Performance, and also the book Alien Agency with MIT Press. I want to agree with Chris Salter that performativity itself needs to be understood as a technical, cultural hybrid form. And tracing it back to the 20th century, in his book Entangled, Chris demonstrates that performative forms and the fascination for the machinic had gone hand in hand. But that in the 20th century, unfortunately, the long history of technological entanglement with performance practice has been ignored or downplayed, not only in theatre and dance histories or in post-dramaturgy, but also in the recent search of writing about the so-called new media. So, along with Salter, we can argue that the career of the term to perform is not only an etymological, but also an epistemological one. And in the last decades, we witness a shift from performance art, from performance as object of inquiry to performativity as a method, while the notion of performance has appeared in the techno-scientific context as well. We have 4 categories I just want to mention. Of course, we all know about this historical reference of John Austin's, who popularized the idea of how to do things this world, the kind of physical, the performative expression and utterance that does not just describe something in language, but actually performs and activates something. Then we all, of course, think of the upcoming gender studies, where the performative program has been pursued by theorists such as Butler, who examines the notion that the materiality of the human body is not given ontologically, but performatively produced in and over time. Then in sociology, anthropology, we have, of course, Victor Turner, the Scottish anthropologist, and theatre director Richard Schechner, and they turn the concept of performance of the subject of research into a method of research. And the performative now allows beyond text to focus on the non-verbal, the embodied, and the immanent act of doing. And this leads us to the 4th category, which is science and technology studies. So where here it takes into account the role of technocultural hybrids, experimental systems, and the agency of non-humans in what French anthropologist Bruno Latour calls human and non-human collectives. Chris has gratified us with this map, and this map is going to give us an overview of what performativity has been evolving as a concept over time. We have been flicking through a kind of great number of agencies in this specific review on performance research, and I don't want to read them all either, but it's quite interesting to see what authors and artists, what have they been dealing with in the last, let's say, 10 years. It can be extraterrestrial organic matter, protocells, artistically created psychotropic microbes, molecules, volatile organic compounds through which plants communicate over distance, of course, DNA sequences, bacteria, fungi, bodily fluids, but also experimental devices such as Winogradsky columns, microfluidic machinery, but also artificial intelligence-based algorithms. And I think I will end here with my short introduction and end with a kind of bon mot by Chris Salter himself. Bacteria perform processes. Scientists perform experiments. Algorithms perform actions. And humans perform gender and sex. The question is who or what nowadays doesn't perform. Thank you.
Thanks so much, Jens. That was just wonderful. And again, I love the confluence of these. Hopefully, thinking about the sort of framework of micro performativity and looking at these networks will be really useful to you. I switched the order around a little bit here so that just to stay on the same computer. So I will let Ray take over now. And not go into any more introductions. Can you hear us all right? And can you?
Yeah, I can hear you. Can you hear me?
Yes, perfect.
All right, great. I wanted to start off by saying thank you all for accommodating me on Zoom. I know as an educator, the last 2 semesters have been in person, but every other week there are students that want to Zoom in. So I know the particular pains of doing a hybrid space, so thank you for having me here. And my project's title is Water Soil Discontent. It's a direct translation from a Chinese proverb, 水土不服. It's these 4 characters. 水 is water, 土 is soil, and 不服 translates to discontent. And I'll go into what each of the elements mean further. Right, so the seed for this project was planted when I saw this paper, which says US immigration westernizes the human gut microbiome. It was published 2018. And they conducted studies among Hmong Thai immigrants that arrived in the United States. And the studies revealed that after a month or less than a month of arriving in the States, their gut microbiome was completely colonized and shifted. And so here's a little illustration of how the gut microbiome diversity decreases over time. And there's a pattern of increased bacteroid strains. And so this got me thinking, because I'm also an immigrant to the United States. I arrived here 6 years ago. And so it made me curious about whether or not my own microbiome reflects these patterns. And so for this residency, I had my fecal samples sequenced for my gut microbiome DNA, and did the same with my mom and my grandmother, who resides exclusively in Taiwan. And if you see here, it's kind of hard to read, but LL, MC, LL is my grandmother, MC is my mother, RH is me. And so over 2 samples each, the bacteroid percentage in my grandmother and my mother were around 50% of their whole gut microbiota. And mine was around 60-ish, so there's like a 10% difference. And so Solon, the lab manager at Coalesce, who has been so helpful throughout this residency, explained to me that this number doesn't just reflect a higher percentage of bacteroids, but it could also reflect less biodiversity of other kinds of bacteria, and hence the higher number in bacteroids. And so this got me thinking 6 years ago when I arrived in the States. Within 3 months, I had developed Hashimoto's disease, and it's an immune disorder where the thyroid is attacking itself. And so I had to go back to Taiwan to receive treatment. And everyone I encountered, from my friends to my parents to the doctor who diagnosed me with water soil discontent. And this term has a long history. The first record of it was in this 3rd century publication called Record of Three Kingdoms. And it's the 4 words that make up the proverb is highlighted in bold. And also in a 4th century publication, the Book of Song. And so I'm going to talk about the kind of first chapter of this project I'm doing at Coalesce, which is a kind of microbiome nostalgia. So with all this information I got, I started wondering if can there be a sort of nostalgia for a gut microbiome that was lost, and does it signal a kind of further removal from not just this sort of idea of motherland, but also from my mother, literally. And the reason I chose my mother and my grandmother as participants in this project and not my other family members is because for me, the gut microbiome has a particular matrilineal heritage. And so my reasoning is that the womb is sterile, so birth is like the first inoculation. When the infant first picks up its first gut microbiome residence, and the breast milk is particularly designed for the baby's gut microbiome. In the non-human, the grooming and the licking is a transference of microbiome. And then in certain insects, the mothers, after they lay their eggs, they vomit this bacteria-laden mucus onto the eggs so that the moment they hatch, the babies hatch from the eggs, they are covered with the right microbiomes. And then in the microbial, in a lot of my work, I work with kombucha and SCOBY, and we always refer to the original piece as the mother and the subsequent pieces as the daughter SCOBYs. And also bacteria's regeneration through binary fission. We also call them like a mother cell and a daughter cell. And so hence begins the journey of collecting fecal samples overseas, over international borders. And so I wanted to go into the process a little bit of how this was done. So this is my fecal sample. And the photo on the right shows kind of the array of agents that went into extracting the DNA from this microbial sample. And so on the left is kind of in progress photo, and the right is just pure gut microbiome DNA. And because I originally work in glass and biomaterials, and so at the same time, I'm doing a residency at Urban Glass, which is in Brooklyn, New York. And during the residency, I began making neon sculptures, and there was an interesting parallel that happened that I wanted to try to cultivate the gut bacteria that was missing in me and present in my mother and my grandmother. But most gut bacteria are obligate anaerobes, and so one was talking about creating this kind of anaerobic chamber to cultivate the gut microbiome. And at the same time, when I was making these neon tubes, they're also anaerobic environments because you have to suck a vacuum and pull out all the oxygen in order to pump the neon gas into the glass. And so I did some experiments on trying to combine or make like a sort of neon biochamber for the gut microbiome. So the photo on the left shows like some experimental gut-like shapes I made with the glass. On the right, some experiments and tests in introducing the medium and the bacteria into the glass. This is a photo of attaching it to the manifold in order to pump the neon gas. And so this whole process has also been a huge learning opportunity for me and for the technician that I'm collaborating with at Urban Glass. And so what we did is we created this little off-center bubble to hold the bacteria so that the bacteria isn't heated up by the process of heating up the glass. And then when the right gas is pumped, this is sealed off, and then the liquid is dumped onto the agar. And so far we were able to get just one of them working. So this is one of them with the bacteria inside with neon light working, because the neon tube is also a sort of sterile environment. So any sort of presence of humidity inside the tube contaminates the tube. And that's particularly interesting to me because ideas of contamination are always in tension with my work with fermentation, which is like creating a sterile environment to have a controlled contamination in a sense. And so that leads to the second part of the second chapter of this project is called Microbial Gyrations. So like Paul mentioned, I came into this presentation with the question of how many citizens are living in this body. And this is a question that was that had started brewing from my previous project. And it's also a response to COVID and to kind of the geopolitics of this origin of this virus and the desire, the anxiety to contain this virus inside a body, such as the bat or the pangolin or the Chinese Asian person. And at the same time, I was applying for a green card. And because I didn't want to hire a lawyer for that, I was going through all the paperwork myself. I had filled out like 200 pages of paperwork. And so that process was also kind of contending with what it means to become a citizen of the United States and kind of what sorts of bodies have the ability to move across borders, both human and non-human bodies. And so I started looking into these movements across lands, and most of this movement happens in the ocean. And so we all know about microbial seas. Most of the oxygen we breathe are created by cyanobacteria in the water, in the oceans. But our bodies are also like microbial seas. We're mostly made of water, and we carry, our gut microbiome has up to like 50,000 different species of bacteria. So we in ourselves are sort of microbial sea. And so I started looking at the ocean currents, which I'm using as a metaphor kind of for these network complex movements. The ocean currents that you would encounter if you were to sail between New York and Taiwan. And that's a pretty rough drawing, but you could see these arrows and lines point to different, like this is the north equatorial current, and this is the equatorial center, the Gulf Stream. And so I began looking at ways to map these movements, and this drawing is inspired by the Micronesian stick charts. So in the Marshall Islands, they have come up with using sticks and shells to create, not map geographical landmarks, but map the ocean currents. And that was in itself a very poetic and powerful metaphor for me. And then that led to me thinking about all of these ferments I'm brewing at home. Comes with this little thing called an airlock, and it's this little device on the left, which is now mostly made of plastic, mass-produced, but they used to be made of glass. And so the purpose of this little device is that in these lungs, you could put water in there, and when the brew is metabolizes sugar and releases carbon dioxide, in a sense, when it breathes, when it exhales, the breath leaves through this pathway and through the water, and then is released. And the water functions like a plug and prevents other cultures from entering the brew. And so I started doing experiments on altering the airlock, on thinking about the airlock as a sculptural form. And so I combined that research with ocean currents and movements. And I created these sculptural airlocks according to the ocean currents that you would encounter between New York and Taiwan. So here are two different ones, a close-up image and an image that more clearly illustrates the shadow, because I'm thinking about these maps, which they were inspired by maps, the stick charts, having also a shadow map when light is shone upon it. And then so that brings me to the last chapter of this exploration, a remedy for water soil discontent. So I first noticed that paper in 2018 that kind of launched this idea because I was already very interested in feces, and particularly in fecal transplant. And so fecal microbiota transplant, FMT, is now being taken up by these biotech companies. And it was it was originally in the literature originating from China in the 4th century, and it was in the record that the name for it was yellow soup. And so I became really interested in this history and this tracing back, because it also parallels kind of that immigration, gut colonization, being traced back to the term water-soil discontent, which also has an origin from China, a very old and ancient origin. So this Ge Hong is the person that first wrote Yellow Soup as a therapy, like using human species as a remedy. And it was also recorded in the Compendium of Materia Medica. It's one of the earlier texts of Chinese herbal medicines. Yeah, and then so this got me like looking into yellow soup and kind of what are its applications in Chinese medicine. And I found that, well, which I already knew, but I never thought about it in a systematic way, because when I was younger and I had eczema, my mom would take me to see a Chinese medical doctor, and I was prescribed silkworm droppings for my eczema. But it was completely normal. I didn't even think about it until I saw this photo, this illustration that was kind of all of the different non-human feces that are within traditional Chinese medicine. And so in the literature, there are kind of 4 types of known human, non-human feces that are used to treat this phenomenon called the water-soil discontent. So there's a little rodent-like animal that only exists in Tibet called Mupin Pika. There's the flying squirrel droppings, which I will have in the performance, but it will not be ingested by participants. There's wild boar feces, which Solon informed me was impossible to get. and then there's yellow soup, which is human feces. And so there's this connection between the water, the soil, and then I realized now that I forgot to explain where the term water-soil discontent originated from. And it's from this idea that when you travel somewhere, even if you're just on vacation, you often encounter these like stomach upsets, diarrhea, gas, fatigue, bloatedness. And the reasoning behind that is that your body is not used to the water and the soil of the new land that you've arrived upon. And so water and soil, and then it leads to soiling, and soiling being a term for shitting in your pants, and then leading to poop. So this line was in my project description, but so kind of what I just presented, taking recent scientific studies among immigrants of gut colonization. This project journeys back to interweave narratives from traditional Chinese medicine and household remedies as an attempt to cut across coloniality, both in the gut and in systems of knowledge. And kind of just like highlighted the words traditional and like asking questions about this idea of what is traditional, and traditional is often paired with primitive or uncivilized, right? And then all these new discoveries in Western science of this gut colonization or of fecal transplants having this root, and then now referring back to this old and ancient knowledge from Chinese medicine, and also how household remedies has already like always been in the cultural consciousness or passed around through oral cultures. And so this phrase is from that Compendium of Materia Medica, and it says that within water-soil discontent symptoms, the most frequent one is diarrhea. And so the feces and its different forms figure into the work as not just remedies in both Eastern and Western medicine, but also as an anti-aesthetic. And so the term anti-aesthetic comes from Alfred Menenhaus and his book, Writing Undiscussed. And the term anti-aesthetic kind of illuminates that disgust was at the center of what we now know as aesthetics. So the philosophical debates around disgust had to happen in order to define what is the limit of disgust, and outside of that is what we call the aesthetic. And in Eugenie Brent-Kimmel's The Form of the Affect, she very aptly points out that when Menenhaus was developing this whole theory around disgust, it took place during the same century of when Foucault wrote his work. And so the disciplining of the body through techniques of power is linked to the disciplining of aesthetics in the 18th century. And then in The Anatomy of Disgust, William, not a direct quote, but William wrote that disgust is a biopolitical tool developed to police otherness. I'm also interested in engaging disgust because of this paradox it contains within it that Colnine writes about. And it's this simultaneous aversion and attraction within disgust. And so that hints at disgust, something being disgusting, always having something behind it. And so I guess, for example, when you walk into a room and there's a foul smell, immediately your body feels disgusted, your face scrunches up. But you also can't help but your mind starts thinking, what is that smell? Or that there's something familiar in that smell, what is it? So there's actually a critical labor of analysis, of unpacking, and of close reading. And that's all happens kind of without voluntary agency. So I'm interested in disgust creating this gap or this possibility, this opening. And then when Menenhaus was developing his theory on disgust, he used the term eklam, which translates to disgust, but it was first linked to excessive sweetness that past a certain point becomes nauseating. And so that's how I designed this pastry ceremony in order to act as this remedy for water soil discontent. So my partner is a pastry chef, and so I think this image is a really good illustration of the aversive connective. This is actually a charcoal puff pastry with a black sesame cream. But I guess with the context of where I'm presenting this could also be interpreted as feces. Actually, when I posted this on my Instagram, a lot of my friends asked me if that was my poop. So within the ceremony, the pastry tasting ceremony, there will be participants who will ingest the items. And because of that, I've veered away from using feces in Chinese medicine as part of the ingredients, and instead use ingredients that are already in gastronomy, but also have a therapeutic effect, like ginger, honey. And a lot of these are household remedies or are remedies in literature about how to kind of alleviate and heal water soil discontent. And so, yeah, it's a long list, but my partner is developing kind of different elements to integrate into this pastry ceremony tasting. And so this will take place at Torn Theater Space, which Paul mentioned. And this is kind of the layout of it. The space is really interesting. It's like a kind of like a typical black box space, but then it has this huge hole dug out from, a huge hole. And then there's a bar in front of it, which is I think it's a Polish bar that is still functioning. And so participants will enter in through the bar and then enter through this tight little hallway space, which I'm conceptualizing as kind of the throat of the space. And when they enter, they'll see the four fermentation tanks with the ocean current airlocks. And then there'll be this huge wooden table with the pastry items laid out. And then within the gut, there's the neon biochambers, and also there'll be a lab table set up because part of the performance will involve lab procedures and kind of delve into the performativity of lab protocols. And then over here is a huge panel of accordion window that will be pushed to the side, and it'll be draped. Instead, this opening will be draped with cheesecloth that has been doused with scatol. So scatol is the chemical compound that makes mammalian feces smell the way they do. It's a result of microbial metabolism. But scatol is also really interestingly used within perfumery, and it's used under 1% as a scent stabilizer, but also it gives off its own like orange blossom floral accord. And so that also illustrates this idea of the paradox of disgust, having the aversion, the attraction with them. And then this
I didn't touch anything. Where are you? Can you hear us? Shoot. Oh, here she comes. Oh.
And this is the window, which will be pushed to the side. And instead, there'll be cheesecloth, and this is the outside, what the yard looks like. I think I'm running over time, but yeah, thank you all so much.
Thank you, Ray. Thank you, Ray.
Paul, you're muted.
Oh. Thank you if you heard me. Oh.
Hit the space bar.
You're still muted.
Oh, thank you, Ray. You hear it? Tell you what, I'll write it in the chat to her.
Dad, there's a new book for.
I don't think it's just a mute. Oh, I see. Can you hear me now?
No, it's not.
Thank you if you heard me.
Hit the space bar.
You're still muted.
Oh, thank you, Ray. You hear it?
Wait.
What? I'm not saying that. I'll write it in the chat to her.
Dad, there's a new book for.
I don't think it's just. Oh, I see. Can you hear me now?
You're still muted.
Oh, thank you, Ray. You hear it? What? Yeah. Thanks for that. Thanks for that. Yeah, thanks for that, Lou. So I think we're gonna move on, and we're really excited to hear what Roland and Julian have to say next. So thanks. Let me, I'm gonna take this down. What's that?
It's not streaming through your laptop, right?
No.
That's really weird. The delay is strange. No, we're cool. We're streaming. We're streaming online from there. From a different computer. This is just the Zoom computer.
Just check the volume. Volume's not on. Just make sure the volume's on. I should be hearing sound now.
I removed this at some point, thinking that this was somehow feeding back.
Dus kun je testen alle sound en curator?
Yeah, that's what I was just doing. It should be running.
This was just working.
See? It is running. Now it should be onto these.
Don't you need the?
That's just for the HDMI.
Yeah.
Yeah.
Might need that.
Yeah. Oh. Do we need this microphone? Let's get a timer. Shall I turn it? Cool. Hey, everybody. Hi. We're going to time it so that we try to stay on time because we both talk a lot. So I'm Julian. This is Roland. Yeah, cool. I won't bother doing an introduction again because Paul did a really good job of that already. That's me. This is Roland. We already established that. So basically, Roland and I first met around 2008. I always forget when we first met. 2018, and we actually first worked together in a collaboration when I was developing this platform that I've got, Teleagriculture at Cultivamos Cultura in Portugal. And at the same time, I was doing, I did some stuff with Roland when he was running the Biohack Academy at Varg. It was quite interesting because when we first worked together, we've kind of progressed from very discrete, separate projects where we're kind of just like doing our own thing and mashing them up together, to then kind of moving to more and more, obviously in this project, we're doing everything together. So this was the first one that we did.
Yeah, and then as for a second one, last year we did the Riverbank Buffet at the Stadwerkstad Out of Matter Festival in Linz, which uses public artistic approaches to raise awareness of food and the environment, using the theme of microbiology. And so we brought together several approaches to food production from around the world in a single menu. And they were all grown along the Danube, many by Julian. This is the menu. We didn't print zines today, but we can give you them later or in digital form. But so we presented this kind of a menu of 10 tiny bites, which all had a small dish, fermented drink, and also an artwork. Presented at the Dierendonck, which is a high traffic area along the Donau, to raise awareness through the public approaches to art, that microbiology is also a catalyst for change and kind of the importance of microbes in our diet. And so every dish had a fermented product in it.
Yeah, so basically these are some of the dishes, some of my food porn. As Roland said, what's really interesting was everything was either grown or caught along the Danube in this very small part of the river in Linz, which is a very industrialized or urban part. And this was up until actually the Nazis in the 40s, a very fertile fluvial agricultural area. So it's quite interesting kind of trying to change perspectives on this and looking at this notion of reversibility. It's also nice to make really nice food because Instagram loves it, apparently. This is the middle one's really funny. This is called Donau duck because it's caught on the Donau, the German for Danube. And it was just the guy was catching ducks and I said, can I have some ducks? And he said yes, so it's quite funny. And also as Roland said, we had, basically we made an artwork very quickly in a week, 10 of them in response to microbial ecologies in these regions and basically us living and participating for 10 days in this area. So that's kind of brings us up to now.
Yeah, so this is our third residency together, now I think about it. And Feel Your Fermented Biome, this project investigates the effect of consuming fermented foods on our identities through the haptification and sonification of the microbial data. Over 4 weeks, and we're now in week 3, these fermented foods are produced, tested and consumed, and their effects we analyze through the microbiome sampling and informatics, so the sampling of our own gut microbiomes, in order to amplify and augment the changes that occur in each subject, so that's Julian and me, through wearable haptic, sonic and visual feedback. That's the dry description. What we in reality do is we have 4 weeks, and each week we have different fermented foods which we consume for kind of a micro-bloating of our guts. So week 1 we did lacto ferments, so kimchi and sauerkraut. Lacto ferments. Week 2, which was last week, we had kombuchas and other acetobacter ferments such as vinegars. Week 3 is this week is natto week. I don't know if you ever eat natto, but it's like fermented soybeans with Bacillus subtilis, which is a soil bacteria. And then next week we're gonna freestyle and we're gonna do whatever our body asks us to do after 3 weeks of fermented food. And every week we take samples of our gut microbiome and send these in for analysis.
Sorry, I'm distracted. So for this, we were kind of undecisive about the fermentation process because obviously this is quite a time-based thing and we are very limited in this project to 1 month. So in the end, I had to do it because I'm kind of addicted to fermenting. So we made a lot of stuff. If anyone, I'm drinking this turbo kombucha. I put 20 scobies in it and it was fermented in 3 days instead of 2 weeks. It's kind of toxic, but really nice. And I've got some kimchi and some stuff if you guys want to try it later. I don't have any spoons. You're just going to have to scoop it with your fingers. But what's really funny was actually because part of this fermentation, yes or no doing is because of the FDA rules and bringing food into the country. This was obviously a decision made for us, but then I actually tried to smuggle some spores in and I became a naughty guy apparently. They confiscated my spores, which is really weird because I couldn't explain that they weren't like some agricultural thing, that they were this food item, and because they lost my luggage, so it was like I wasn't there when they were inspecting it. And then, anyway. So that's a funny story.
Another aspect of this project is beside the food is that Julian and I actually live together for 4 weeks, which is perhaps even a bigger challenge than the diet. So we're cohabiting, in fact, in the same house in Buffalo. And Roland, the lab manager of Coalesce, sent us this article last week on the Dutch microbiome findings. So they did this huge study with, I think, it says here, more than 8,000 Dutch individuals, and which is funny because I'm Dutch, right? So this might also be my microbiome, but they showed that they researched the factors that shape the healthy gut microbiome, and they found that it's primarily shaped, which is blue here, by the environment and co-housing. So co-housing was in fact a much bigger factor in the microbiome than food. Which brings us to the next thing, which is our own co-housing situation. There's one thing to know before I start our little documentation of where we live, namely that we live in this kind of Airbnb, which is a split house. So we both kind of have our own house and our own temperature system. So we live together, but also we have a kind of separation between us, as you will see.
Press again.
This is our mornings.
Morning routine.
So there we go. As you see, we all go to our own living rooms. So the thing is that Julian always likes it much warmer, and I'm always too warm at night. So we have this thing where I always...
Luckily, we have our own thermostats and heating systems.
Yeah, because he's from the desert in Australia. Well, then of course we go to the toilet. It's very important for the project. So this is our Poo Sampling Kit. And we decided to not do it for the video.
Did you flush the toilet?
Did I flash it?
Gross.
Oh, there he is, yeah. Yeah, so this is us literally every morning.
Yeah, so it's interesting actually because we kind of we're working in these very kind of interesting scales, you know, at either end of this, like very domestic approaches and very kind of pseudoscience of things like mindfulness, and then these very, you know, high-end like DNA 16S sequencing and all this kind of stuff. And so it's quite interesting. But in terms of the domestic journey and this kind of looking at microbiomic assimilation through living together, we also developed this kind of this system in the morning of guided meditation that I was kind of doing anyway before this. Then journaling, personal journaling independently. Then we meet up together to do a morning check-in, which was a video logged event every day. and then eat breakfast. And it's kind of, yeah, I find this kind of interesting, this kind of like oscillating between very like domestic kind of science and very kind of high-end lab science. And yeah, so this is my favorite meditation person. I do guided meditation, and she tells me that I'm worthy and that like I have everything I need every morning. And then you do the mood, which was Friday. I always get stressed because I realize I haven't done enough shit in my week, so that's my Friday. And then this is one of Roland's poems, right? Poem? Journal, yeah. So that's our kind of workflow in the morning, every morning, which is why we take like 3 hours to come into Coalesce. Get in at like 11 every morning.
Yeah, and what we also do then at the end of that is like a video check-in, which is kind of meant as to this kind of season 1, 2, 3. So every morning we start to say, hey, this is episode 1.1, and we record a little conversation to check in how we feel and to see how the diet affects us. Today is going to be our last check-in of the series because it's a presentation day. So this is our final check-in. We just want to show you how it functions.
So we're going to check in for you guys.
Yes, we're going to check in for you. So it always goes like this. Like, hey,
Usually I get a coffee though.
Oh yeah, it always goes like this. Yeah. Today is Friday, 22nd of April 2021.
3.5.
Episode 3.5. And I say, Julian, how you feel?
I feel good. I feel weird doing this in front of a lot of people. Yeah.
Yeah.
How do you feel?
Yeah, kind of like every morning. Yeah. How does the current diet affect you?
It's the worst week for me.
Natto is where.
Natto sucks eating it every day. Like I've not eaten this much natto before in my life, so it's an adjustment. But yeah,
Any other thoughts you want to share? Yeah.
no.
So I really miss kimchi.
Which is funny because he hated kimchi in week 1 and now he's addicted to it. Like,
Yeah.
I'm like the heroin dealer of
Because
kimchi.
I had 100 grams at lunch and dinner, and now I feel when I'm in the shop, my body craves it, but I can only do it next week. It's funny. It also produces serotonin in the brain.
But I've made 10 kilos of it for next week. So.
Anyway, let's continue because now we're
Yeah.
going like a barely routine.
We're at 10 minutes, dude.
Yeah, good. So, as for the lab stuff, we did fecal sampling, DNA extraction, which is now sent in to the sequence facility, and
We could do one.
and we also did some culturing in the lab, but the culturing is more as a side project to also be able to see some of our microbes, to see if they're actually in the samples. We also did a thing where we did culture the microbes and kind of put our shit together, basically. And little microscopy we did with gram staining the bacteria to visualize them. But the real data that we're working on is the 16S data. So we sequence this part of the genome, which can show you which can show you which kind of microbe it is. So we send all this information in, and we hope to get a lot of data back about the different microbes. And this is our spectrometry data, which shows that we actually have DNA in the samples.
So this is getting into the kind of more bioinformatics side of things. Basically, we got introduced here to this platform, this software called Genius, which is genius actually. It's amazing. And what we really liked about it was we get this kind of like overall snapshot of what we're looking at. If you've ever looked at like a sequence, even a 16S, which is quite specific, it's still very hard to kind of get your head around how it works. And this is a really nice way of kind of doing some calls and some searches and kind of just kind of going, that's different. So this is kind of a tool that we've just learned that we're adopting for this project. But I'm just going to go through my kind of pipeline of bioinformatics now. Obviously, like we said, we're in week 3 of this project, so it's a work in progress. But basically, what I've been doing is I've been looking at sorry if I nerd out a little bit, but whatever. I've been looking at doing like databasing of the 16S sequences and querying it in SQL with PHP querying to kind of like sort through it and build more, as you can see here, of a like taxonomy. From that, I kind of realized really quickly if I wanted to kind of produce a workable prototype for you all to look at today and think we're cool, I needed to adopt a different strategy. So what I ended up doing was I ended up developing a whole discrete set of JSON tables and basically simulating these spikes that we are hypothesizing that will occur. So for example, there's a static data set for Roland and I of our microbiome growth, which has a kind of an algorithm which is kind of implying growth because we only have one sample to work with. Then going through and basically separating us out, and then separating us out to kind of simulate the effect of like like as you can see for week 2 in Roland, like separating out like particular orders and of genuses of bacteria that we both would expect from microbe loading those particular foods to increase and decrease as the experiment continues. So then from there, I started thinking about how to kind of process these microbiomal fluctuations. And so just because I'm okay at it, I decided to start working with Python for a number of different reasons. One is that it's quite easy to do a growth simulation. Another one is that it's also quite easy in the long run to start doing stuff with machine learning for predicting outcomes and for basically what we're doing, thinking about it over the 4 weeks to kind of do a longer longitudinal simulation. Yeah, and then also I'm porting it all out into OSC, which converts the data, if you don't know, into a WAV file, which means we can use it for vibrations and sounds and all kinds of cool stuff. Okay, so what I wanted to do, thinking about all of this, because data kind of does my head in a bit, I wanted to do a visualization of this to get my head into what this was. And it's really funny because my dad always says you can't polish a turd, and that kind of remember this because we have a lot of shit conversations about shit. And what I wanted to do is I wanted to make shit alchemy, so I wanted to turn our shit into gold somehow, because we kind of, we're kind of mixing his shit with my shit and kind of, trying to make this beautiful art project out of it. So this was kind of basically looking at 2 different forms of data and kind of, trying to map that into a 3D space. And what I realized was just by mapping it onto the vertices of a 3D object, that this is way more complex than I was thinking. And because we're looking at building empathy and looking at producing emotional responses, we decided to simplify it. So then I built this thing in processing. So basically, this is both of our microbiomes. It's just a sphere. The idea is that it's kind of like the sphere is the original baseline sequence, and then these kind of weird things that you're seeing happen are basically happening for our spikes in different bacteria in the sequencing. My idea for this was also to kind of, I really like this idea of kind of like touching immateriality and things like this. I did a project a while ago doing this with dark matter and infrared interaction. But so the idea would be that you can kind of touch this so you get a sense of agency over your microbiome. So the things are happening and we're kind of not controlling the microbiome is, but then we also have an agency over that, obviously, which is what we're doing with heavily eating food. And then I then patched all of this genome data into PD to make a kind of pretty abstract sounding, arbitrary sounding synth. So basically, this is me and this is Roland, and our microbes are singing to you, which, should I demo it now or at the end?
Yeah, demo it now. We do the haptics at the end then.
But we do it all at the end.
Oh, we do it all at the end.
Yeah.
We demo it at the end.
Yeah. Okay. So Roland's going to talk about haptics now.
Right, there's another part of the project, which is called Feel Your Fermented Biome, and it's also part of my kind of PT research strategy, which is all about sensing microbes with haptics, is that we create different haptics prototypes which will be wearable, and these haptics have, maybe you can already show one if you want to demonstrate kind of quickly this one. So every thing we have now is like a breadboard with 4 little vibrating motors, and then these 4 points can actually convey information, because you could write with 4 points, or you could create different buzzes or kind of increases in vibration or decreases in vibration. So we are prototyping with libraries made specifically for haptics and for feeling this vibration on the body, and thinking about how to map this microbial data into buzzes that you feel, which we now do in Arduino a lot. So this is me the whole day looking at which kind of buzzes and transition ramps and signals are interesting, and then testing that in the it makes sound too in the little thing. So you have this little vibrating motors which on your body will give information about the microbiome. So you can also feel your own microbiome, but more importantly, maybe I can feel Julian's microbiome. So this kind of creates also empathy between us, because you don't only feel your own microbial data but also those of others. And we're looking at different kind of haptic integrations and next steps. Yeah, we're almost done.
Yeah, we don't need to talk about it. So obviously, like I said, we're at the start of this kind of processing the project and making it into art now. So we've got a lot of shit to do still,
Yeah.
literally and figuratively.
And now we end with a very short demo. You want to do sound too?
Yeah, I'll just get this set up.
Yeah, you get it set up. So
Where's the mouse?
this is like the breadboard version, right? But the idea is that I have now Julian's microbiome data, and I'm wearing this whole thing the whole time, so I'm going to show you. Originally, we want to make a wearable, so now, because we don't have that yet, basically we kind of, paste it on the body. So this is like the little device, which now gives me information about Julian's microbiome. I wanted to say something more about it, I don't know if we can stay here for you. But in general, what we're working on now is, because we had this conversation with Paul as well, like the difference between arbitrariness and chaos and kind of signal and randomness. So how can you make this data interesting in a way that I can actually sense the microbiome and know what it means, or doesn't it matter, and can it be arbitrary, and can we still kind of sense something?
Yeah,
So
I
that's
think
what
this
we're
is,
working.
yeah, this is very much in that line
Yeah.
between representing it as an idea, but not quite specifically enough. And there's lots of,
Yeah.
you have these decisions, you know, like do I put all the names of genes here and map that? So does that become
Yeah,
educational?
what
So
does that buzz mean, or that buzz?
Yeah,
in the end, the
yeah.
question is whether like these microbes are travelers, or whether they take residence in us as the artists in residence, like microbiome residence in artists. Which is beautiful.
and it sounds like it's meteorite, right? That's what it sounds like. Yeah, meteorite.
This is live, yeah?
Yeah, this is our microbiomes. Yeah. Roland's on the left.
Beauty.
Yeah. So I'll just turn that off now. It's probably enough of that. And we would like to just say, yeah, obviously we didn't thank you. So thanks for coming. It's really nice, obviously, to do a laser talk. It's actually my second laser talk in a month, so I'm like a Leonardo laser guy now. Yeah, and yeah, it's obviously really great. And thanks for this, Paul and Coalesce. Yeah.
I have to say, this is already, I think, shaping up to be the most fascinating BioArt in the public sphere we've had. So I'm really excited. Tosca and Andre. Yeah. Oh yeah, sure. I can stall for you for a minute.
Okay, well, what I was
I'll
going
let-
to do, I think, is show some slides.
Okay,
And then,
sure. Okay.
I don't know. Hopefully it's gonna work. Hello, I can't see at all. Uh-oh. Doodle-doo. Hi, everybody. Oh. My notes so I don't tangent too much. Okay, I wanted to say happy Earth Day to everyone, and in my opinion, every day is Earth Day, or it should definitely be looked at as such. So I thought I'd give like a brief overview of my new media and immersive work that I've been kind of doing in the 21st century. So I've been a professional metalsmith for over 30 years and a glass artist for almost 20 years. I've always really been interested in computer-generated graphics and things like this since the late 80s, so a long time, and integrating that into my metal work and later into my glass work. I should say, like, I'm originally from San Francisco, California, and I left the United States in 2001 for Canada. So I understand to a certain degree what it's like to relocate to another country, and for myself going through that whole process was fairly interesting. But so fungi has kind of been my main focus now for almost 10 years. But before that, I have been kind of creating my other worldly explorations. And here I'm showing some work that I presented through Urban Glass Brooklyn, where I created like my own pathogenic kind of fungus. And Andre, my partner, who's together we call ourselves Nanotopia, but I'll go more into that in a bit. We have always kind of worked on soundscapes and things like this together. So I show this work. This is one of the installations I did where we really started integrating soundscapes into those environments, kind of as a way of what that world would sound like. So these are all metal and glass. And around 2011, I really started questioning the materials I was working with, particularly metal, even though I was recycling a lot of it. And glass work takes a lot of energy just to be able to melt it. Like I tend to cast glass and I blow glass and things like that. And it takes a lot to do so. So fungi, substrates and things like that, I thought I could sculpt with. It was just an interesting thing that when you add certain kind of nutrients and things like this, particularly I found like psyllium husks or xanthan gum mixed with brown rice flour, which brown rice flour tends to be something that's integrated into substrates to cultivate mycelium. But with the xanthan gum, it not only acts as this incredible nutrient, like the mycelium loves it, but it kind of makes the substrate more clay-like to be able to manipulate. And so because sound has always played a big role in my work, I started wondering, what does mycelium sound like, or what do these organisms sound like? And I started creating different circuitry. Here I'm showing my monophonic moss piglet, which didn't allow me to listen to the mycelium, but I could touch, I don't know if you can see my mouse there. Yeah, okay. So if you touch this, it works kind of in the same methods with conductivity, and it would make this horrible buzzing sound. So also alongside that, like researching different circuits and things like this, I was sculpting larger mycelium sculptures. So here this shows in 2019 a large piece I did for Grow Up. It's about 1 meter tall, and people could come and kind of create a duet with the mycelium. And another myco-material exploration was with the Mycelium Martian Dome project, where it was actually kind of part of a collective called the Alien Agencies Collective, which Chris Salter talked about, and it was with a combination of all of us, Joel Ong, Nicole Clouston, Elaine Whitaker, and Gunish I forget. Eastman. Yeah, sorry. Sorry, Gunish. So while we all created our own works that showed at the Ontario Science Centre, our main theme was kind of space. And so I was thinking about like domes created out of mycelium. I chose this geodesic dome structure. And within the negative spaces, some of them I grew the mycelium directly within it, and others I made panels and placed them inside. And within, you can kind of see it here, the alien, if you will, like the Martian, lived within there and created a soundscape within the space that people could interact with. And so over the pandemic, a lot of things, as everybody knows, went online and things like that. So Forest Undersound was installed at the museum in Kitchener, Ontario. However, I mean, it lived there by itself with a lot of other artworks because the museum and everything had to shut down. But what I did is with hemp, I created a substrate, contained it within these special like terrariums, and here I did like a cardboard culture. And over a certain period of time, like maybe it was 3 months into this project, I heard from the janitor there that the mycelium had escaped the container. And here, like it's fully sealed, melted, welded together. And actually, I should say, I kind of, they were worried gases would build up and the thing would explode. And I said, there's no way that's going to happen. But instead, what happened is the mycelium fully cultivated that substrate and did what I didn't expect it to do, because there was air coming through this front portion and kind of here, where these are just plants from the Ontario, like kind of native plants. But there's electrodes that come out here and go into these synthesizers. So I figured there'd be enough air, the mycelium wouldn't want to escape. So I'm going to share here with you. I believe it should work. The day I had to come in and kind of rescue the mushrooms that escaped the container and the sounds that they were producing. Oh, why did that happen? Sorry, let me go back. I tried to click this. I don't know. There we go. I don't know if I can't make it. Can you, do I need to plug sound in somewhere? Or?
Yes,
Sorry.
on the table.
Oh, okay. Sorry. There you go. So I started working with these purpose-built circuits that I call Biomodules. And what they do is they detect microfluctuations in conductivity between 1,000 to 100,000th of a second, and translate that in real time or quantize it through Arduino code, and then put that out, like output it through MIDI or control voltage. So I can take that information into synthesizers or into different applications to have the fungi in real time disrupting visuals and things like that. And it got honorable mention. So another thing I did was the Mycorrhizal rhythm machine. And so with that, the concept was that I would take a space 10 kind of structure, and over like a lot of these works, I'm interested in how the sound changes, or like grows more frenetic or kind of dies out over longer periods of time. And so the idea was that these beautiful herbs and everything here is a mycorrhizal kind of symbiosis that happens, where the mycorrhizal fungi grows within the roots of the plant and helps the plant generally be able to withstand like different temperatures or dryness in soil and things like that. And people could come within the dome, and of course they'd hear the sounds everywhere, but they could also put their hands onto touch plates that would be going through this mycorrhizal fungi. And again, kind of, I'm interested in people collaborating, if you will, with non-human. But unfortunately, the gallery attendants didn't water the plants, and then some other people overwatered the plants, so all of the plants died, which I guess would have happened eventually, but my hope or kind of vision was that they would thrive, and I would return and there would be this jungle of herbs that people could collect from and share and things like that. So it is, for me, it's been really interesting working with living organisms and that whole kind of how you approach galleries and things like that, because they can either thrive or they can die out. So for, in May 2020, I was part of the New Nature Climate Science Exchange that was hosted by the Goethe Institute Montreal and curated by Samara Chadwick. So there, a lot of artists, scientists, and technologists came together to present different works on what a new nature after the pandemic might look like. And so for that, I was really interested, as I've been with fungi, but how we might become more empathic and have more compassion towards non-human, especially like in the forest, like we don't see, mycelium unless there's like fruiting bodies of mushrooms and things like that. And I thought it might be interesting, because I was meeting with all these technologists and VR specialists, what if we could go within the mycelium network in like a VR scenario? And so I'm showing this, just a brief kind of aside. So something that sprung out of symbiosis dysbiosis, which is, I started that in May 2020. And last year, some of my co-collaborators, we were thinking it might be fun if we, because it's such a long kind of process to figure out these different microbes and fungal networks and things like that for a simulation, what if we create an environment where we could perform in it for a Maze Berlin festival, and Mycelia came out of that. And so Mycelia resides in VR Chat, which is a social VR platform. And so there's like fully tracked dancers. I embody a avatar in there that looks like I'm made out of fungi and things like that, while I'm sending biodata of mushrooms into that VR Chat environment, which is audio reactive. And people could touch my avatar, which is connected to haptic sensors that are placed around the mycelium. So it's like acting as a form of touch. So when people touch me back in my studio, these sensors vibrate and the mushrooms respond, which people can hear in the soundscape or see visually, which we're going to do a little performance here for you soon. So these are some of the incredible people that are collaborating with me right now. And unfortunately, the neuroscientist, Brendan Lehman, couldn't come up, so we could have people come and try on some of the tactile sensors we're working with. And some other people that worked with me on Mycelia are based in South Africa, and other people in Berlin. And so it's been like this really interesting kind of connecting with all these people around the globe. And Alison Moore, who is shown here, is a Montreal-based new media artist, and she's been collecting photogrammetric and LiDAR scans of British Columbia coastal rainforest, like tree areas, and I'm collecting boreal forest region. So we're creating these point clouds out of this photogrammetry, which to me, point clouds really have a microbial look to them. And I like that. So you can touch these things, and my idea is, I'm just really curious, like what we leave behind or what comes with us. Like, when you touch a tree, does some of that biome come onto you? What do you leave there? And so the photogrammetry works with that. And then I'm going to show you this video, and then the next slide, I'll talk a little bit more. So these are some of the photogrammetric scans that then I've put through and done some things in Touch Designer with, so you can see more of these, like kind of the idea of the microbiome through the forest. And the sounds that you're hearing, it's from actually an EP that we put out, but a lot of it is mushroom mycelium based, where we haven't really interacted with it at all, other than maybe reverb or delay. Saturna Island. So part of the VR experience is the mycelial interface, and here I'm showing the purpose-built circuit biodata board, and there's also a company, like I may as well say, we're working with the Eurorack synthesizers, so there's different modules. If you're unfamiliar, like you can customize and build your own synthesizer that way. And there's a company, like the biodata module, actually, let me back up, is designed by an electrical engineer called Sam Cusumano of Electricity for Progress. And he's made all of this data totally open source, so people can work with this, expand upon it. Like, he's very interested in that. And he worked for a company called Data Garden, and he designed what's called the MIDI Sprout. And there's some history there, but he has kept his schematic and his code open source for people to be able to use. So one of those companies or people that worked with it is called Instro, and they made a special module based off of his, and I have one in my rack. So tonight, that's what I'm bringing data into the synthesizer with. But so we're also working to kind of try to create a more one-of-a-kind experience for each person that goes within the VR. I'm working with neuroscientist Brendan Lehman, who I mentioned, and we're using the OpenBCI ganglion to take EEG readings. We're interested in how we might combine the Earth's Schumann resonances, and here's my note on that, if you're unfamiliar. So Schumann resonances are a group of natural electromagnetic earth energies. They're important to humans because they help induce brain states that help people relax, heal, and perceive and experience earth energies. They're a set of spectrum peaks in the extremely low frequency portion of the Earth's electromagnetic field spectrum generated and excited by Earth's surface in the ionosphere. So like lightning and things like this. And what we've noticed is just similarities in the mycelium biodata that comes in and certain brainwave frequencies with the Schumann. So what we're thinking is that when these align, there's different things that will happen within each person's experience. It will be a little bit different, perhaps how, the trees are responding and the microbes are moving around. And we're also working with technology called the Emotibit. So that allows us to also kind of look at emotions people are feeling through sweat, heart rate, and it's also like movement, so how people move within the space. And so I'm here at Coalesce to expand further into that mycelium network portion of the VR experience with scanning electron microscopy, and confocal, which I've never worked with before, but I'm very interested in that, and atomic force microscope. So with the atomic force, an idea I have is looking at the frequencies because everything vibrates at the atomic level. And with scanning electron microscopy, due to the nature of how that looks, I can turn that into bump maps and things. So without further ado, we're going to do a small performance here, myself and Andre. And the mycelium, I'm about to mention that. So I don't know if I need to unplug. Like, I don't know if you've got the picture. But so there's mycelium right here. There's actually huge containers and plates full of it at the lab. Solon has been very kind. And so it's sending its electrical energy. It's very active right now. Sometimes it gets quiet, but a lot it's active right now. So, yeah. There you go. Mycelium is the vegetative part of a fungus or fungal-like bacterial colony consisting of a mass of branching, thread-like hyphae.
we went a little bit over what we were thinking today, so I thought if I thought maybe just if there were a couple very sort of brief questions or something, we could have them, and hopefully if we answer them, we'll try to answer them very relatively briefly. But thank you so much for coming out. This has been awesome. I think this has been one of the most fascinating of the presentations so far. I mean, it's like just the amount of like, I'm just thrilled by the amount of like gizmos and tools everybody brought out, and this has been really cool. It's been awesome. I would normally I would get Ray back up, but I'm almost thinking it might be better just to, since you all are here, maybe just to focus the questions on you guys, and we'll have to take questions for Ray later, right?
I'm just like close to the computer, I'm sorry, unless you want to keep that in.
Oh yeah, sure.
Sorry.
Sure. So if there are, maybe if does everybody want to come up, and and we can take maybe 5 minutes of questions or something? If anybody has,
Like we go up?
yeah, I was thinking if you guys come up. I'm thinking, Jens, Tosca, Andre. I think we might. I don't want to keep people here super late at but I thought if there's a couple burning questions, we should really take them on while everybody's here. Nice. Reinhardt.
Paul?
Yes.
No, I'm just curious. Sorry I came in a tiny bit late. I'm totally fascinated by the mycelium and that whole world. But I was really interested also in the sound sources and a little bit more about programming and the things that you're using, the systems you're using to sonify. Can you expand on that a little bit? Yeah.
So what I'm doing is I have electrodes placed within the mycelium. I'm using wire. And lately I've been working with acupuncture needles, and Solon, who's amazing, has asked me if I've considered like micro needles. I'm like, micro needles? So I think he's going to help me try to get some of those, because we're thinking maybe we can insert within a high flow thread. So they must be like incredibly small. And so they're going into a biodata that detects microfluctuations in conductivity, again, between 1,000 and 100,000th of a second, and it translates that in real time into MIDI notes and controls or control voltage out. So I can take that data because of MIDI, musical instrument digital interface, and plug it into synthesizers because it's a protocol. Or you can use it like in different applications like Touch Designer or Unity. It's pretty fantastic. But with control voltage too, because we're working with Eurorack synthesis, is we can take that and like 1 volt per octave, we can go into different modules and trigger them with gates and things like that. So it's creating pulses. And so like the biodata boards, not necessarily what I have on my Eurorack, but it's quantizing them into different scales. Like I can create my own scale and take these fluctuations and change them. And often when I'm touching, like earlier today, when we set up here, it was super quiet. Like there was almost undetectable data was flowing into the synthesizer. So I had to keep touching and kind of moving the mycelium to have some information, like some of that electricity come through.
Extension of that question, is there anything microtone happening, or is it all equal temper?
Well, with the synthesizer, Andre.
Yes. Yeah, I mean,
I know.
the interpretation scale is admittedly ours.
Yeah.
But one thing we have observed working with different mycelial culture, things like that, the more you work with it over time, you start to recognize pattern. You don't recognize necessarily pattern on a human scale. If you record MIDI data, you can look at it and look like a punched piano roll, and you can visualize patterns that might be longer than your attention span listening to something you haven't heard before can actually recognize.
So you're making it familiar rather than necessarily microphone where people's tonality.
Yeah,
and so on.
admittedly, that is an interpretive function.
Okay, cool, thank you.
I have a question with with all the kimchis and the
Yeah.
cottages,
It's us.
the microcosms. So you were doing some great visualizations with your work. Do you foresee that you're going to have those visualizations in like a mobile form? Because it seems like it would be really good to have like this haptic feedback for people nearby you that you know. As like I was thinking, like if there's something going on with like the biometrics of somebody that they don't need to verbally tell you they need something, you kind of just give them what they need to like settle out their system. And I was just wondering, like with your outputs, if you're going to make it something that is not like so gallery or installation based, but something that's just on the go.
Yeah. It's a fantastic question. I mean, the work is partly inspired by research done in the field of haptics, where people study this type of devices, and especially in like the deaf-blind research community, or kind of researchers who make health devices for deaf-blind people. This is a big topic. To make, how can you convey like the alphabet or information or signals in like 3 or 6 vibrating discs on your body? I don't know how we're gonna develop it. I don't know yet, but I'm also interested in kind of, for example, if I could feel something about Julian that he doesn't even know yet that he felt, right? So it's also some kind of like subconscious gut feeling, literally. So I don't know if I'm only interested in kind of the literal thing, but also in kind of the unconscious bit of like me knowing you.
Okay. To answer your question, which you asked, Roland, yeah, I mean, so one of the really nice things, obviously, by, you know, because at the end of the day, there's database, like it's really static data, you know, it's not like evolving in real time, and it's an interesting question from the point of view of it's also very expensive to send away these 16S sequences. You know, it's like we got them for $30 sending them to Canada. There's $70 downtown in Buffalo.
Yeah.
So there's that cost factor in that sense. But in terms of a mobile version of things like the visualizations and things like that, sure. And that, what I was going to say is because we don't live in the same place. It's an interesting possibility that evolved out of me trying to build it in a telematic way. So basically, we will continue working on it, not living together, and we'll be doing everything online. So yeah, I don't know if I'm answering your question either, sorry. Basically, something mobile could come out of it, yes.
Next iteration probably going to be a wearable that you don't stick on your body.
Are you still wearing it?
Yeah.
Yeah.
Hey, want to see that?
No. The weapons? Yeah, cool.
Any other questions?
I have a question.
Yeah.
When do you start to think of these agencies engaging in a collaboration, and when do you think that you are abusing them or instrumentalizing them?
Am I abusing the mycelium? I have felt as a collaboration, and I know there's a lot of question in that. I feel, if anything, for myself, I feel very protective over what I'm cultivating. And I know a lot of other people are now starting to look at like plants and different things like that, at least as far as like this similar type of circuitry. And I'm not wanting to work with it as like a novelty or anything like that. I found that a lot of the sculptural pieces that I do and the music that people are hearing, of course they anthropomorphize and things like that, but I've found people, like at one of the slides I showed with the large sculpture, there was a woman that was crying, like she was just really just broke down, and for her it was just this realization that she felt she'd never really considered, you know, like things that you don't see. I had this with a child too that couldn't see the plant growing, so he thought it was dead, like he didn't consider it like a living thing. So if for me, I guess, I mean, it's definitely something I think about a lot. And I don't just, like, I don't ever throw things in the trash. I put them out in the garden under a tree, or I've buried things where slime mold have attacked fungi and stuff in hopes that the fungi will spore and slime mold, will go away. But I feel it's bringing empathy for some people, like just considering the non-human life as life, if you will, like that these are living systems and
Not
things like that.
life with an F, not just with an V.
Yeah.
Yeah.
We're literally flushing ours down
Yeah.
the toilet like all the time. So, yeah, I don't know how that is in terms of agency. I guess we're distributing our people love our shit. But yeah, I mean, I think it's interesting in terms of control and agency in that point, because when we think of human entanglements, we're always thinking of them as still us being this dominant party. That for me is one of the interesting things about microbes and microbiomes, is because we kind of almost like at their mercy, and like, I think, Ray talked really well about all of this and how it is a system of control and a system of cultural dominance and things like this. And I didn't mention it, but it's interesting for me, having lived in lots of different places, the kind of like rhetoric around probiotics and microbes, and like here it's very like kind of fitting into this kind of like, I would call it a paradigm of the left, whereas in Austria it's kind of like everyone does it, and in Australia, and so this is kind of, in terms of public engagement, an interesting kind of challenge the perceptions of these little bacteria that make up so much of this. But in terms of control, I think it's quite interesting as well, because Roland has a much more, I would call it, conservative and controlled diet. I'm smashing my body with fermented shit like all the time. And so for me, I'm actually having a lot less. And it's a weird thing, because I'm feeling like I was drinking 2 liters of kombucha a day before I started here, and then I had to just go like dry for a week, and I was like really like jonesing for some booch, and it didn't happen. Yeah, but so that's interesting, because you do get this weird reliance, but then also you don't know if that's just a psychological thing, and that's a lot of our project is like that, if it's just in our heads, basically.
It's an interesting question. I mean, I've heard this question a lot of times, but now I'm thinking about it in our specific project. And with this gut microbiota, I mean, we are hosting them. Anyway, so we don't try to kind of change our gut microbiome into anything it isn't. Although we do eat like lacto-fermented food, but then these can take host in us, or just use it as a highway to go out again on the other side. But sorry for that metaphor. But I mean, it's completely different because before I did, for example, a project more on biodigital games, which are the steers in the organism, which is much more like a kind of control paradigm. But in this case, basically we are also kind of listening to our microbiome in a way. So it's more like what Julian says, like what they are telling us in a way. Yeah. Which is interesting question.
I'll
of
say I've
agency.
noticed since being here, like coming to Buffalo, just a short distance from Toronto, whoa, like everything is totally changed, and I've wondered if it's got to be the water. Like I've had that when I lived briefly in Iceland, and definitely that sulfur water and everything really knocks you. But coming here, like I've had some stomach stuff, and I'm like, is it food poisoning? Is it the water? What's going on? And
The other side
I don't
of
think
Lake
it's
Ontario.
any of that. Yeah. I'm like, what's yeah.
Well, we should thank you so much
Yeah,
for
thank
coming, everybody, and really appreciate all the questions. And if you have if you're interested, get in touch. My contact information's on the end of this poster. And again, thanks again, and have a good weekend.
Sorry, that took me longer. I was curious about your notes.
Yeah, that's all.
Yeah.
That was amazing.
Actually, Wednesday.
Ah, sorry.
That dye, or that that dye.
Food.
So we have pizza back at the house.
Oh,
Okay.
Sam.
Oh, okay.
That's where Stephanie
Well, you don't actually have it.
Well, I have it
She went to
here, but
intercept the
the module I'm using
Yes.
is here,
So she's
and now
back
it's
in our
not plugged into
Cool.
this on.
Okay, we have to collect this stuff.
Yeah. We have time to
Quite
collect it.
Yeah, it's called Scion, so it's based off of Sam's.
Yeah, we could use it pizza delivery. I mean,
Yeah. No, I
Also,
think
I think I'm
everybody's
gonna
gonna
get this
starve
tomorrow.
by the end of
You
this.
cannot access
Yeah, I figured, you guys have so much, like, stuff, so much, like,
So it's particularly
media, you know, like, so much stuff. And so it's like, we had, it's gonna be,
So then,
Yeah, it's cool, yeah. We felt very punished.
So very much. Yeah, that was a nice touch. Very stellar.
Like I said, it's weird.
Yeah. We originally wanted both to have one, and then
that was it.
Yeah,
Yeah, it was a real stellar moment.
very sticky.
Yeah, good. Well, we have pizza and wine.
Yeah, no, I meant sticky.
Oh, yeah.
I'm like,
Yeah, we've got pizza and wine back in us. Stephanie and the boys went back to get here's that was that dramatic moment of tea swapping.
Yeah. I didn't see that.
20.